So now we get to check out the sheep brain. So again, the structures that you're going to be required to know on the sheep brain are on the handout. It's a shortened list of structures. So I'm sure you're happy to hear that. And while this looks a little bit messy to begin with, hopefully as we continue through the dissection, all will be revealed. So what we're seeing here is we are seeing some additional structures that are associated with the brain. So you can see some of this is adipose tissue. And we can see also even some cranial nerves that are passing through. There's going to be some blood vessels and things like that. Remember that the sheep itself is oriented horizontally, whereas we're going to be oriented vertically. So whereas our cerebrum is going to be up here, and then we saw the midbrain and the brainstem and the cerebellum kind of coming down like this at a right angle, for the sheep, it's going to be coming off like this instead. Okay? And one of the things that we're seeing that's kind of obscuring our view of the brain is this right here. So this white that we see right here is called the dura mater. So remember we saw the dura mater when we were looking at the spinal cord models? It's the durable outer meninges, if you will, the membrane that helps to protect the brain. So there's the cerebrum that we see underneath right there, and this is usually covering it and protecting it. So this dura mater, we couldn't see it on our brain models, but we could see it actually on the sheep brain like that. So if it's pointing to this, that is the dura mater. Now the pia mater, remember, is the innermost one. The pia mater is going to be like saran wrap right on the surface of the brain. So if you were to kind of get, we can see into some of the sulci here, if we were to get just inside here and kind of lift up, see how it's kind of catching there on a little membrane? That is going to be the pia mater. So it's that delicate that we see right there. So see, it's kind of lifting off like that. So that's the pia mater. So the dura mater is the durable one out here. The pia mater is the one just right on the surface of the brain itself. A couple other things that we can see. We're now looking at the inferior surface of the brain. So here is our spinal cord down there, and then our brain stem is going to be through here. In this area here, see how between my thumbs right there, there's a bulbous structure. It kind of looks like a pea. That is the pituitary gland. So that's the pituitary gland that hangs off the inferior surface of the brain. This is the optic chiasma, where the optic nerves are coming in and joining and then forming that X that we talked about. But you'll notice that the dura mater wraps a lot of the cranial nerves, wraps the pituitary gland, etc. So I'm going to have to take off the dura mater so that we can see our brain structures, and likely a lot of this is going to come with it. So that's why I wanted to highlight it ahead of time. But you can also see how the dura mater goes in between the two cerebral hemispheres as well. It dips down into that longitudinal fissure. So we're going to work on removing that. I'm going to get my head in the way of the camera. We're going to peel that out of the way. So there's our longitudinal fissure that we see there. It kind of dips down into that. So we're going to move that. Things a little bit more slippery. Now we can see the reveal of our underlying structures. So again, you can see some of the nerve pathways going through that dura mater like that. And there's our pituitary. So our pituitary gland is right there. It's embedded right here. So we're just going to remove the whole thing like that. But it's still embedded within right there. Okay, so now we can see a view of the brain somewhat similar to what we were seeing on the model, right? But again, our spinal cord and our brainstem is coming out at an angle like that instead. So we can see our cerebrum. So this is going to be our frontal lobe here, our temporal lobe, our parietal lobe, and then our occipital lobe. That longitudinal fissure that I was talking about is going right along here between the two cerebral hemispheres. So it goes right along here and kind of separate it like that. moves through our pia mater, etc. And so we're going to do a mid-sagittal cut right through that. We're going to continue on through the inferior surface of the brain. Okay, so again, now we're getting a view that is somewhat similar to what we've seen on the models. There should be, my particular example here, for whatever reason, the one that I picked up, doesn't have a good cerebellum showing. So our cerebellum should be back in here. So along with this could certainly, this is part of our cerebellum right here. So it's on the posterior part like that. Okay, so this is one half of our cerebellum, and it'd be right associated with our brainstem like that. Okay, other things that we can see is this is our corpus callosum right there. In the sheep brain now, we can actually look into it. That's giving us a view into our lateral ventricles. And you can see some of the vasculature in there. That's some of the choroid plexus that we have right there. But it goes deep inside here, just like you saw in the ventricle model. So that's the corpus callosum. And this area right here, in here, is going to be our thalamus. So our thalamus is going to be the kind of the rounded area that we see in here. The hypothalamus would be just underneath it. It's not quite as large in this particular example, but this is going to be the thalamus, the round area, and the hypothalamus is going to be underneath it. And remember that pituitary gland was going to be a little bit darker glandular structure underneath that, but it's separated with our dura mater there. Other than that, we can also see back here, this is part of our corpora quadrigemina. So our corpora quadrigemina is going to be located on the posterior side of the midbrain, and there's going to be 4 bumps. And if we look back here, look, there's a bump there, superior colliculus, and then there's a bump here. So those are the 2 individual out of the 4 bumps, superior colliculus, inferior colliculus, that are making up that corpora quadrigemina. You'll also notice, see that little dark structure right there? That little ball that you get right there, which technically if you were coming in here and viewing from the posterior side, we can see how it kind of separates. That ball that we see right there, that's the pineal gland. The pineal gland that releases melatonin that we said that works to coordinate our sleep and wake cycle with day and night. So that's our pineal gland. That was our corpora quadrigemina, that's our thalamus. As we work our way down here, this kind of anterior bulge, it's a little bit more subtle on the sheep, but this anterior bulge that we have here is going to be our pons, and then just below that is going to be our medulla oblongata. And as we continue down, that would be our spinal cord down below. Sometimes, depending on the cut, looks like I was a little off, we know that our third ventricle is going to be here, in between the 2 halves of the thalamus. And sometimes you can actually see the cerebral aqueduct kind of coming down and going into the spinal cord. So if when you do your cut, your mid-sagittal cut, if you do it right in the middle, you should see a little groove coming down here. Because remember right in here, and again, mine was kind of falling apart, there's our cerebellum. And the 4th ventricle occurs between the cerebellum here and what we see as the rest of the brainstem, like where our medulla is and our pons. So this area right here is going to be our 4th ventricle. You can see there's a little indentation there. That space right there would be our 4th ventricle. And if sometimes you can get the cerebral aqueduct, you'll be able to see a groove leading down to that. Okay, so let me look at our list. Dura mater, pia mater, gyri and sulci. We know that the surface of the cerebrum has all these wrinkles to it, right? So it has these hills like this, and then it has valleys down below, right? So the valleys are called sulci, and the hills, if you will, are called gyri. An individual one is called a gyrus, or an individual groove or valley is going to be a sulcus. So again, it's like cactus and cacti, sulcus and sulci, gyrus and gyri. So you're seeing the plural in the list there. Longitudinal fissure, we said, occurs between the 2 cerebral hemispheres. So again, that opening that we see there. Transverse fissure, again, a little bit messy because my cerebellum is kind of a bit of a mess. But there's a transverse fissure. There's a gap here between the cerebellum, and the cerebrum. So that gap that we see here between the 2 is called the transverse fissure because it's more horizontal. cerebral cortex, the cerebral lobe, cerebellum, pons medulla, corpora quadrigemina we did, pineal gland, olfactory nerves, and bulbs. Here's the inferior surface of the brain. You see these here? Those are basically the olfactory bulbs as well as the nerve pathway is going to continue through. So those that we see here, right? Sheep do a lot of smelling of things, etc. So olfactory sensation, smell are going to be routed through these nerve pathways here. So those are the first cranial nerves right there. Then optic chiasma. The optic chiasma was kind of hard to see in my particular one because of the way that the dissection was coming. It was coming off in there, but it was going to be this X-form structure underneath there. So we can't quite see it on my example. We did thalamus, hypothalamus, corpus callosum we saw. Corpus callosum through there. And then the pattern of white matter inside of the cerebellum, you'd have to cut into it in order to be able to see it. So you can see a little bit. See how it's dark here and it's light there? So that's part of that arbor vitae that we were seeing. So myelinated neurons through the white matter, unmyelinated neurons in the gray matter. You can also see that in the cerebrum. So if we do a parasagittal cut, right, mid-sagittal right through there. So we do a parasagittal cut or just a sagittal cut that goes in parallel with it like this. Then what you're going to be able to see is, again, see the pattern of gray matter on the outside and then white matter on the inside. So those are where the myelinated areas are versus the unmyelinated neurons and axons there. What else do we see? Cerebral peduncles we talked about. Mammillary body, I wouldn't worry about mammillary body. Cerebral aqueduct, we couldn't quite see it in mine. And then fourth ventricle also would tend to be here. It's going to be just anterior to the cerebellum. So that's part of what you get when you go to an actual, organ, is that it's going to not be perfect like the model. But most of those structures that we saw, probably 80% of the structures on the list that we have there, we could actually see on the sheep brain itself. So on the lab practical, we would expect that you would have a pin with a number on it. It might say, name this structure right here. That would be our thalamus. Or it might be name this white structure right here, or corpus callosum, or name this lobe, or something like that for frontal lobe, etc. So only the things that we have on the list from the handout would we be required to identify on the sheep brain on the lab practical. Okay? But it's a very straightforward dissection. You're just doing that mid-sagittal cut along the longitudinal fissure, removing any of the dura matter. Yours may or may not have the dura matter associated with it, but certainly be familiar that this leathery piece here is the dura matter. Okay? So when we're doing the dissection, you want to make sure you don't have to do the dissection. You can be at your table the person that watches the dissection, but you have to be able to identify those structures. So you're going to need a tray and a brain, a scalpel, and then some sort of a probe like this. We have blunt probes over there. And do the dissection, identify the structures, and then when you're done, there's a bin on the ground there that has a red bag lining it. So it's a hazardous material container. All of these, since they're preserved materials, need to go in there. Any pieces and parts, little bits and pieces like that, have to go into the container. So the halves of the brain, any of the pieces and parts go into the container, and then rinse your dissection tray. You can also remove this insertion here, this little insert, and kind of rotate it 90 degrees like that so that it can dry. So you're going to rinse it all, but make sure no pieces are going down the sink. And then when you set the trays, you can stack up the trays and take that insert and rotate it 90 degrees so there's a little bit of air that gets in there as well. Okay, don't worry about it if they're still a little bit wet, or just allow them to dry over the weekend. Okay? Alrighty. So when you are ready, go grab yourself a brain, an extra brain there, and we can do our sheep brain dissection.